Howdy, Pima Aztecs, and welcome to part 3 of chapter 9, controlling microbial growth in the environment. These are the chemical methods that we use to control microbial growth. So check out this cartoon. So clearly, this is bacteria in a fedora and a trench coat, and this is another bacteria. And he's got this. The guy in the fedora has got this piece of DNA, and he says, "Hey kid, want to be a superbug? Stick some of this into your genome. Even penicillin won't be able to harm you." So what he's doing is he's saying, here, take this piece of DNA and a plasmid, stick it in your DNA, and then you can make proteins that will help you evade penicillin. And it says here, it was on a shortcut through the hospital kitchens that Albert was first approached by a member of the antibiotic resistance. So this is pretty funny. You've probably heard of superbugs. You know bacteria can become superbugs. But as we go further into this course, this cartoon will be even more and more funny. So it says here in this bullet, Americans are overly preoccupied with antisepsis and disinfection. So you can get your soap, your shampoo, your toothpaste, etc., all of these that contain disinfectants. And there's little evidence that all these items that we put disinfectants into, there's little evidence that adds to our human health. But what it does do for sure is it promotes the development of resistant bacteria. So the more we try to aimlessly kill bacteria, the more resistant they become. And if they become resistant enough, they can become a superbug. So now back to microbial control chemical methods. So here's some Clorox. Probably everyone's seen that. Betadine, it's usually used for surgery, and here's isopropyl alcohol. So chemical methods are used more often to disinfect things. Chemical methods are used for antisepsis and also for food preservations versus the physical means, which was the previous lecture. If you use a chemical method to control microbes, those methods can adversely affect cell walls. So you can disrupt their cell walls, their cytoplasmic membranes. So remember that phospholipid bilayer. Chemical methods can disrupt that. Chemical methods can disrupt proteins and DNA in bacteria. Chemical methods and their use are affected by the temperature. We looked at that last time. Oftentimes, temperature, if you warm up or heat the chemical that you're using to kill a bacteria, it's more effective than that same chemical at room temperature. Chemical methods are also affected by the length of exposure. So in the lab, in our micro lab, if you spill some media and it's got microbes in it, what you want to do is put a paper towel over it and then soak it in 70% ethanol and then wait 15 minutes and then wipe it up and then clean the area. So the time of exposure of the bacteria to ethanol would be 15 minutes. Also, chemical methods are affected by the contaminating matter. So, is a contaminating matter vomit, or like I said, some culture media with bacteria in it. pH is important in deciding which chemical method to use. The concentration of the chemical method. So because alcohols evaporate so quickly, you don't want to use 100% alcohol solution as your chemical method. You'd want to use 70%. And also freshness of the chemical. So in the lab here at the U of A, we change the 10% bleach every week. Chemical methods can be used to destroy every living thing, even endospores. So that's pretty cool. There's 9 categories of chemical methods. There's phenols, alcohols, halogens, oxidizing agents, surfactants, heavy metals, aldehydes, gaseous agents, and enzymes. And we'll go over all of those in this lecture. Here's a cartoon of a phenol. So it's a 6-membered double bond ring with this OH or hydroxyl group on it. Oh, here's some hydrogen peroxide. I bet everybody has some of that in their house. So if you use a chemical method as a sterilant, that should kill 100% of the microbes. If you use your chemical method in the form of a disinfectant, that's going to kill less than 100% of microbes on surfaces. If you use your chemical method as an antiseptic, that's going to also kill less than 100% of the microbes on the living tissue. And then a sanitizer will remove microbes through cleaning them off, so basically just wiping them off, not necessarily killing them on site. So let's look at phenols and phenolics. So again, here's a phenol, and then B, these are all phenolics, and then these guys are bisphenolics. So you can use a phenolic as an antiseptic and a disinfectant. And this guy, triclosan, which is a bisphenolic, and here's its chemical structure. So again, it's got the hydroxyl group and it's got a couple chloride ions on it. Oh, it's a phenolic compound. Commonly it's been added to soap, but this raises concerns about selection for more antimicrobial resistant bacteria, so maybe we don't need to put that into soap. Probably everyone's heard of Lysol. Phenol is the active ingredient in that. And this guy, Joseph Lister, used phenolics in surgery when he first made popular these antiseptic techniques. Bisphenolics, you just have 2 linked phenolic groups, and we already talked about this. Triclosan is the active ingredient in Lysol. What it does, though, it can denature proteins. So again, if bacteria has an enzyme that it needs to do metabolism to live, and that enzyme is a protein, and triclosan destroys the protein, then the bacteria can't do the biochemistry and metabolism, and it will die. Phenolics can also disrupt the phospholipid bilayer, the cell membranes. So that was phenolics. Now the second chemical group that we're going to look at to control microbes is alcohols. And here's a photograph of isopropyl alcohol. So when you swab your skin with alcohol, it's more of a degerming method. Doesn't necessarily kill the microbes. And we talked about this, 100% alcohol will evaporate too quickly to kill the microbes. So you should use 70 to 90% alcohol. Alcohols are used for both antisepsis and disinfection, but alcohols aren't used to sterilize things. Alcohols will denature proteins and disrupt membranes, just like the phenols will. But alcohol is not effective against fungal spores, and alcohol is not effective against endospores. And this is a cartoon up here of denaturation. So let's say this guy, this green thing here, is a enzyme, and it's made of protein. And what alcohol will do is to denature or make that protein enzyme fall apart, and now the enzyme can't work. That's denaturation. And I don't know if you've heard of these things before, tinctures. They're just alcoholic solutions of antimicrobial chemicals, a tincture. So the next group of chemical methods to control microbes are the halogens. And if you've had chemistry, you know the halogen, you know where the halogens are on the periodic table of elements. But for this class, the chemical category of halogens contains chemicals which can be used as disinfectants, sterilants, so a sterilant is just something used for sterilization, and also antiseptics. And they include the halogens iodine, chlorine, bromine, and fluorine. Iodine, used as a tincture or an iodophore, it's a long-lasting, non-irritating, and it's non-toxic to humans. It's an oxidizing agent. So oxidizing agents get reduced. That means they take on electrons. So, and then the molecule that lost the electron got oxidized. And we'll talk more about oxidation reduction reactions in this lecture in a few slides. Betadine or betadine and povidone are both halogens, and they're commonly used in hospitals. I guess scientists aren't exactly sure how they work, but they could damage enzymes. So like if they're protein enzymes, they get denatured. The enzymes become denatured. Backpackers used to have these iodine tablets to treat their water when they go and they drink from a creek. Just put the iodine tablets in it. But it's better to filter your water because those protozoan cysts can survive. And here's a betadine or betadine being used to prep a hand for surgery. So here's the hand getting prepped for surgery, scrubbing it with betadine. More halogens, chlorine. So here's Clorox. It's got chlorine in it. It says kills 99.9%. So chlorine, chloramines are used in wound dressings, for skin antiseptics, and you can use it in drinking water. So it says down here, 2 drops of bleach in 1 liter of water can be used in an emergency to make the water safer to drink. Not ultimately safe, but safer. Chlorine is non-carcinogenic. It's non-toxic to humans. And bleach is actually sodium hypochlorite. So here's the chlorine component of bleach. And this is used as a disinfectant. So bleach is a halogen that's used as a good disinfectant. And probably everybody's been swimming, so they know. Swimming pools, you can smell the chlorine. More halogens to control microbes. Bromine or bromide. It's used in hot tubs because it evaporates really slowly. You can also use bromine, aka bromide, in swimming pools. Fluorine. You probably know that you have fluoride. It's a residual chemical in your toothpaste, and it's there to reduce the number of cavities or dental caries as it disrupts biofilm. Okay, now I promised we would talk more about oxidizing agents and redox reactions. So here's our cartoon. We have an electron donor, so that's someone's going to donate the electron, and an electron acceptor. Someone will take the electron. So just as a quick review, let's look down here on the left-hand side. So this is an atom, and if you remember from 201 or whatever biology class you took to learn this, you've got the protons and the neutrons here in the nucleus, and then the electrons, which are negatively charged, circle around the nucleus in electron levels or shells. So here's an atom. We're going to call it A plus 2. That means it's overall this molecule, this orange molecule, has an overall plus 2 charge. And it's got an electron. So once it donates its electron to molecule B, that means that molecule A has lost one negative charge, so that means it's going to become more positive. So the orange ball gets oxidized and becomes molecule A3+. So when it loses an electron, it got less negative. That means it got more positive. That means it went from a +2 to a +3 charge. The green molecule, on the other hand, the green molecule B+3, means it's got a +3 charge on it. It took an electron on, so that means it became more negative, and that means you went from and it's just one electron, so it went from a B+3 to a B+2. So that's what's happening in the world of electrons. So the electron donor is called a reducing agent, and the reducing agent that donates the electrons gets oxidized in an oxidation in the oxidation part of the oxidation reduction reaction. And then this guy, this is the oxidizing agent, gets reduced because it got an electron and its charge got reduced. So this is a little confusing, but it's weird that this green molecule got something, but then when it got something, it got reduced. That feels weird, seems weird, but that something that it got was this negative electron charge. So the +3 charge was reduced to a +2 charge because it took on a negative charge. And then, so this guy that got the electron got reduced and became the reduced acceptor. Okay. These reactions, these redox reactions, which I just talked about, are going on all the time in your body. And if you're giving up an electron, you always want another molecule to be there to accept the electron. But if that's not the case, then what you get are these things called reactive oxygen species, ROS. And they'll have unpaired electrons. So here's molecular oxygen from the atmosphere. That's fine. That doesn't have any unpaired electrons on it. But all these other reactive oxygen species have unpaired electrons. Unpaired electrons are dangerous because they can rip through things in your cells, including proteins and DNA, and cause a lot of trouble. So you don't want these reactive oxygen species with their unpaired electrons roaming around your cells. So here's some of them. Superoxide anion, this red one, there's the unpaired electron. Peroxide, this is part of hydrogen peroxide. That's got these 2 electrons that are unpaired. Hydroxyl radical, there's the unpaired electron. And then here's the hydroxyl ion that doesn't have an unpaired electron. And then this cartoon just shows, this is generally true for humans or bacteria. There's usually kind of this sort of balance. There's going to be a reactive oxygen species in your cells, but you'll have these antioxidants, like a lot of fruits and vegetables have antioxidants in them, to battle the reactive oxygen species. And you can see it's just kind of a nice balance. If you tilt the balance toward the reactive oxygen species, you get a disease. If you tilt the balance toward the antioxidants, then you're a healthy person. So we eat lots of fruits and vegetables. So we talked about this, the oxidizing agents get reduced. That means they get an electron. And when they get reduced, they're oxidizing microbial enzymes. And that, if you are oxidizing microbial enzyme, you're messing with its electrons in the atoms in the enzyme, and then that enzyme can fall apart. If the enzyme falls apart, you're inhibiting microbe metabolism because the enzyme has lost its function, and then the microbe can be destroyed that way. So hydrogen peroxide, here we go, hydrogen peroxide. It's not good for superficial wounds because in our skin, we have this enzyme called catalase that inactivates hydrogen peroxide to water and just molecular oxygen. But if you have a deep wound where the anaerobic microbes live, so they don't like oxygen, they don't like hydrogen peroxide, because they don't have an enzyme, they don't have catalase to detoxify the hydrogen peroxide. Hydrogen peroxide is good for disinfection and sterilization of contact lenses, but that's the only thing I know that it's good for, and then deep wounds. Some high level, you can also use hydrogen peroxide for some high level disinfectants and antiseptics, depends on the percent. So if the hydrogen peroxide you're using is less than 3%, it's an antiseptic. If it's 3 to 20%, it's a disinfectant. And if it's 25% or above hydrogen peroxide concentration, it can be a sterilant. So here's what I was talking about with catalase. So if you are a microbe living on the skin, you have all the enzymes that can detoxify reactive oxygen species, including catalase and peroxidase. If you are an anaerobic microbe that doesn't like oxygen, living deep in the tissue, you don't have these enzymes. And when you put hydrogen peroxide on those microbes and they don't have catalase to detoxify the hydrogen peroxide, or they don't have peroxidase to detoxify the hydrogen peroxide, then hydrogen peroxide will it will turn into a reactive oxygen species, this peroxide ion with these free electrons, and will destroy the bacteria. But if you are a microbe, an aerobic bacteria, you do have catalase, you do have peroxidase. You put hydrogen peroxide on those bacteria, they just detoxify it with the enzymes catalase and peroxidase into water. Here's a catalase test that we do in the lab. Catalase will convert hydrogen peroxide into molecular atmospheric oxygen and water. So the top slide, this is Enterococcus faecalis. So it doesn't have catalase and Enterococcus, so it lives in your gut and in your feces. So it doesn't have catalase, so it can't turn, and so if Enterococcus faecalis is put here on this slide, and then you put hydrogen peroxide on that slide, Enterococcus faecalis doesn't have catalase. It can't turn hydrogen peroxide into water and oxygen, molecular oxygen, which you would see as bubbles. Down here, however, this is Staphylococcus epidermidis. So you put Staph epidermidis here, you put hydrogen peroxide on it. Staphylococcus epidermidis has catalase, so it's catalase positive. So that means Staphylococcus epidermidis is going to change the hydrogen peroxide into water and oxygen. And here's the reaction. So Staphylococcus epidermidis has catalase. You pour hydrogen peroxide on it. It turns hydrogen peroxide into 2 water molecules plus molecular oxygen, which you see as bubbles, as oxygen bubbles. More oxidizing agents could be ozone, used by Canada and the EU to treat drinking water. Peracetic acid you can use to sterilize surfaces, and it's sporicidal. Surfactants, soaps and detergents. So here you are, washing your hands. Boy, we know how to do that nowadays. Everybody's washing their hands, which is good. Soaps and detergents are collectively known as surfactants. Soaps are good degerming agents because you use friction to degerm your hands. Soaps are poor antimicrobial agents unless the soap contains a specific antimicrobial agent. They physically remove a microbe from the surface, through friction, like I said. Surfactants found in hand soap have a hydrophobic lipophilic and hydrophilic lipophobic structure similar to the phospholipid bilayer in the cell membrane. But the hydrophobic ends of this hydrophilic structure break up oily deposits, and the hydrophilic ends attract water molecules. And the result is that the oily material and the bacteria are washed away. Surfactants generally are effective at removing microbes due to their ability, again, to disrupt cell membranes, just like we saw with the other chemical means, especially when you combine a surfactant with a mechanical friction or rubbing, which is what you do when you wash your hands. And then detergents are also surfactants. They're positively charged molecule. They're a little more soluble in water than soap is. Okay, more surfactants. What about these guys called quats, quaternary ammonium compounds? They're commonly used detergents. They're harmless to humans. They disrupt the membranes of the bacteria again, but they're not effective against non-enveloped viruses. They're not effective against mycobacteria. They're not effective against endospores. A famous quat is benzalkonium, but these guys are also quats. Okay, how about using heavy metals as a means to control microbes? So here they are. Here's some heavy metals. Arsenic, zinc, mercury, silver, copper. They're all antimicrobial because they combine with sulfur atoms in proteins to denature them. So these are disulfide bonds. So these red things are disulfide bonds. So they use sulfur to give this green molecule its shape. When you add heavy metals, the heavy metals cause these bonds to break, and the protein denatures. So it denatures into this thing. Now this thing's not functional. This protein could be an enzyme and it's not functional. So we said enzymes can be protein. I don't know if you've heard of this Silvadine. It's topical, so you put it on your skin. It's for burns. It contains 1% silver. So you don't, when you get a burn, you don't want bacteria to colonize where the burn is. Silver creams or a silver cloth is also good to help control microbes. In the olden days, they used to use silver nitrate in newborn babies' eyes to prevent blindness that would be transmitted by Neisseria gonorrhea in the mother. So coming through the vaginal canal during delivery, if the mother has Neisseria gonorrhea, the babies can become blind because of that microbe. Okay, microbial control, enzymes in general. Here's a famous one, lysozyme. So your tears, your sweat, your saliva all contain this enzyme called lysozyme. And it's pretty cool because it digests the peptidoglycan cell wall of bacteria. Prions, it's been hard to eliminate them. You have to incinerate everything. And by the way, that's what a prion looks like. But you used to have to incinerate things that came in contact with the patient that had prions. But now there's this amazing new product called Prionzyme. It's an enzyme that gets rid of prions. Okay, more heavy metals. How about mercury? This is this mercury compound, thimerosal. It's used for 70 years to preserve your vaccine. But because mercury is a metabolic poison in 1999, they tried to do less vaccines preserved in thimerosal. Mercury is also in some of your dental fillings. Aldehydes, more chemical means of microbial control. Aldehydes are capable of sterilizing. So aldehydes are thus called sterilants. Aldehydes will cross-link proteins that will denature them, and if you're denaturing or destroying the proteins in the bacteria die. Aldehydes can also inactivate nucleic acids like DNA and RNA and kill bacteria. And some examples are glutaraldehyde and formaldehyde. So here's a protein. It's got its protein shape, and you use an aldehyde fixative. You get these cross links in the protein that can destroy it. And then here, this cool old-timey picture of, I don't know, looks like the 1800s or something, of a mortician embalming a dead body with formaldehyde. And they put the formaldehyde in water and they use it to embalm people. Formaldehyde is really irritating to mucous membranes and it's carcinogenic, so we don't want to use that a lot nowadays. How about gaseous agents to kill? So it says here, fast and simple, 100%. ETO sterilization. So gaseous sterilizations, they're the most toxic to humans. So if you get a job and they tell you to go run this gas sterilant machine, then you should definitely know how to use it, because if you don't, it's toxic. So gas sterilants include ethylene oxide, propylene oxide, beta propiolactone. So like I said, when you get into the clinic, you'll probably learn all these words. They're highly reactive chemicals. They're highly toxic. But you can sterilize plastic laboratory ware with gaseous compounds. And you can't put the plastic labware into an autoclave. You can also put in heart-lung machines, sutures, mattresses, pillows, artificial heart valves, etc. And put them all in these machines and sterilize them with gases. But make sure you know how to use the machine. Here's some facts on gaseous agents. The gases rapidly penetrate into every crack, whatever you're trying to sterilize. They denature proteins by cross-linking. We've seen that. They use ethylene oxide a lot in hospitals. Gaseous agents, however, are highly explosive. They're extremely poisonous. And they're potentially carcinogenic. And then here's a list, a table of chemical methods of microbial control. So we went over all of these, phenols, phenolics, alcohols, etc. So just remember that a chemical agent that dissolves lipids, like in the phospholipid bilayer of the bacterial plasma membrane, can also damage cells and envelope viruses. So if you're a chemical agent and if you can dissolve a lipid, you can damage the phospholipid bilayer of the bacterial cell. And remember, the envelope viruses just what they do is they when they left your cell, they took some of your phospholipid bilayer from your plasma membrane with them, and that made them an enveloped virus. The end.